Before mitosis occurs, we have interphase. During interphase, the cell carries out its regular metabolic functions and prepares for its next division. These are three phases in interphase: G1, S, and G2. During G1, important growth processes are occurring. The cell grows quickly during this phase. About midway through interphase, the cell's DNA is copied exactly. That is, the DNA in the chromatin replicates to create a second identical set of DNA. The final phase is G2. DNA replication in the S phase has consumed a great deal of energy, so this second growth stage lets the cell rebuild its reserves of energy to prepare for division. Prophase is the first of the four phases of mitosis. During prophase, the chromatin condenses into tightly packed chromosomes. One pair of cylindrical organelles called centrioles move apart to opposite poles of the cell. As the centrioles move apart, a network of fibers called the spindle apparatus forms between them. The second phase of mitosis is called metaphase. During metaphase, the spindle fibers guide the chromosomes to the equator or centerline of the cell. The spindle fibers attach in such a way that one sister chromatid faces one pole, while the other sister chromatid faces the opposite pole. The third phase of mitosis is called anaphase. During anaphase, each centromere splits apart and the sister chromatids separate from one another. The spindle fibers that link the centromeres to the poles of the cell shorten. As these fibers shorten, sister chromatids are pulled to opposite poles. The fourth and final phase of mitosis is called telophase. Telophase begins when the chromatids have reached the opposite poles of the cell. The chromatids begin to unwind into the longer and less visible strands of chromatin. A nuclear membrane forms around each new set of chromosomes, and a nucleus forms within each new nucleus. Like somatic cells, germ cells proceed through the growth and phases of interphase before dividing. This also occurs before a germ cell begins meiosis. In prophase 1, each pair of homologous chromosomes align side by side. This aligning of homologous chromosomes is called synapsis. Although they contain the same genes, the homologs carry different alleles of these genes. A pair of homologous chromosomes is made up of 4 chromatids and is called a tetrad. While the homologous chromosomes are lined up side by side, non-sister chromatids may exchange pieces of chromosome in a process known as crossing over. Following prophase 1, a spindle fiber attaches to the centromere of each chromosome. A spindle fiber from one pole attaches to one pair of sister chromatids in the tetrad, and a spindle fiber from the opposite pole attaches to the other pair of sister chromatids. The chromosomes, however, do not line up in a single file as they do in mitosis. Instead, they line up as homologous pairs. During anaphase 1, the spindle fibers shorten. This causes the homologous chromosomes to separate from one another. The homologs move to opposite poles of the cell because the sister chromatids are still held together. The centromeres do not split as they do in mitosis. In telophase 1, the homologous chromosomes begin to uncoil and the spindle fibers disappear. The cytoplasm is divided, the nuclear membrane forms around each group of homologous chromosomes, and 2 cells are formed. Each of these new cells contains one set of sister chromatids and is now haploid. The phases of meiosis II are similar to the phases of mitosis. During prophase 2, new spindle fibers form, nuclear membranes disappear, and the chromatin condenses into chromosomes. Metaphase 2, the chromosomes line up at the metaphase plate, and chromosomes are made of 2 chromatids at this point. Anaphase 2, the centromeres split and individual chromosomes move to opposite poles. Telophase 2, chromosomes arrive at poles and the cytoplasm starts to pinch off. Each cell that enters meiosis II is haploid but consists of replicated chromosomes. At the end of meiosis II, the daughter cells are still haploid, but they contain single unreplicated chromosomes.
It looks like an Asian, Eric Cartman, you should respect.